So our third panelist is Saskia de Vries. She's the Associate Director of Data and Outreach at the Allen Institute for Neurodynamics.
Great. All right. I'm excited to kind of continue this conversation. I think this is a really important topic, and it's been very exciting to listen to the talks over the last couple of days and to see the range of different types of approaches and techniques and modalities that this community is engaging with. So, as Stephanie mentioned, I'm here at the Allen Institute and the Allen Institute for Neurodynamics, and there we go. I won't go into details about the science that we're doing. We're not doing things that are nearly as cool as what you guys are doing, but we're trying to understand how the brain and the circuitry of the brain and the activity of the brain drive behavior, learning, and decision making. And we do this with cell type resolution, but at a brain-wide scale. And so we take a number of different approaches that includes electrophysiology using neuropixels probes, chronic electrophysiology, a variety of different behavioral contexts, calcium imaging, fiber optometry, as well as a lot of neuroanatomy, usually using expansion microscopy and light sheet imaging. So there's a lot of different types of data that we're working with that we're trying to bring together in order to answer some of these fundamental questions. So my role is largely to create our data platform that we can use to organize and analyze our data as well as share our data. And this platform has a number of features requirements. And the first is that it needs to be multimodal. And I put this little illustration on the side. This is from a recent review of other work that has happened at Allen in the last decade of studies that have really tried to integrate information across different modalities in order to understand the circuit and function in the brain. And this is a lot of the underpinning of our work, is we need to link transcriptomics with physiology, with anatomy. We need to bring models into mesoscale connectivity. And so we need a data platform that allows us to both collect and analyze and process these different modalities of data, but also be able to integrate them together in a meaningful way. Our platform needs to be flexible, again, to support diverse experiments and analyses, but also in the long run because we wanted to support the diverse reuse of the data. Supporting reuse and encouraging reuse, I think, is one of my primary missions. And what I'm kind of most interested in, I love that Ben was able to reproduce these grid cells, but I don't want you just to reproduce my grid cells. I want you to be able to use the data that we're collecting to ask different questions of the same data that is in the community. And finally, we want a platform that's collaborative. Within our own teams, we need to be able to collaborate, and we want to be collaborating with the broader community. At the end of the day, open science is really about collaboration. So the approach that we've taken is to build a cloud-based platform that allows us to store our data in a single place and work with our data in a single place and to bring people to the data rather than distributing data to the people. And so if we think about 3 of our scientists on our own team that all work on different things, one's an electrophysiologist, another does anatomy, and the third one builds models, all 3 of them are going to deposit and work with their data in this common cloud infrastructure. This is really useful. Even in the same building, it's often very hard to work with data across computers. Particularly, some of these data sets are very large, and so even if you're sitting next to each other, that can be a challenge. And so having the cloud really minimizes that challenge, or reduces, I should say. But it also is really effective when we're trying to collaborate with people who aren't in our own institute. When we're trying to collaborate with people at other institutions, either across town or across the country or in other places, it's very easy to give them a link to access the cloud rather than having to send them a hard drive or try and put things in Dropbox and do all these things. And so if we want to then be collaborating actively with some of our colleagues in other locations, bringing them into the same cloud platform makes this a lot easier. But then at the end of the day, we want this now to be accessible to the broader community. And so again, having this in that one cloud location makes it easy to open this up to the rest of the science community. So this is the kind of the foundation of our platform. Just to kind of go through it in more detail, some of the things that we need to have a platform that supports community collaboration. We need the data to be accessible. This means that all parts of the workflow are openly available. So this includes raw data, derived data like analysis results. It includes the analysis code, metadata that goes with the data, protocols that were used. All of these things need to be accessible. They need to be standardized. Data is immutable and conforms to standards, very similar to what Ben was just speaking about. We want the data to be transparent so that the data and code are documented. That facilitates understanding what it is and being able to reuse it. And then finally, we need our work to be reproducible. And this entails building containerized compute so that when we build analysis workflows, we're able to reproduce them and share them with other people that they can reproduce them as well. So the way that we've built this, I've already mentioned, we're putting our data and our work in the cloud. Our data goes in a bucket on AWS. Our code is all going into GitHub in public repositories. And then we've got an organization or a page on protocols.io where we document our methods. We lean into the community data standards, so NWB for our physiology data and OME for the light sheet imaging data. Our data is transparent, and I'm going to dig into this a little bit more in a moment, but we're establishing a metadata catalog that documents the data as we collect it, as well as tools and portals for visualizing and exploring the data. And then finally, as I mentioned, we need these reproducible workflows that for our analysis work. And for this, we're leaning into a platform called CodeOcean, which is a cloud-based data analytics platform built around these types of containerized compute. So I'm going to talk really briefly about our containerized compute and then a little bit more about our metadata. The need for this reproducible analysis is that we need to understand, after we've collected the data, we need to understand the processing that happens to it. So with electrophysiology, we do our recordings, and then we do a whole bunch of spike sorting and signal processing and unit quality assurance. And we need to really be clear about how we're doing that when we share this with other people. And we need workflows that we can share with other people to use with their own data so that they can kind of coexist side by side. To kind of illustrate this, you can imagine, so for like calcium imaging, you've done your cell segmentation, you use Suite2P, and your methods section in your paper says we use Suite2P to process our data. This is very similar to this instruction on how to draw an owl. You start with 2 circles, and then you get this owl, right? It's not that this is a false statement, but it really leaves a lot of information hidden about how you actually went from your raw movies to your segmented cells. And so we need pipelines that actually document kind of all of the preceding steps up until Suite2P, all of the parameters that you set when you run Suite2P, and then how that's being transformed and used. So we've been using CodeOcean. I mentioned again that this is a cloud-based data analytics platform. It's built around this concept of capsules, which are containerized compute. What that means is that you link specific data and code with environment. It doesn't use any, it's not using any black magic. This is using GitHub for code. It's using Docker for the environment. But it really facilitates the ease of doing this so that my scientists don't have to learn how to write a Docker file. They learn how to use a little GUI to specify what they need in their environment. And now they have a capsule that they can share with their neighbor. They can share with their colleagues across the world. That's always going to run the same way because that environment is fixed. It also allows us to track easily the provenance of data. So when I've run some data through a capsule and I've saved a result from that run, it shows me which capsule was run, what was the data that were inputs to that capsule, and then I can even explore this further and click on things and find out the parameter settings and things like that. And then finally, we can link these capsules together to create a pipeline. And what this means is each step of this pipeline, each step of this workflow, it might be something like you're doing drift correction on the electrode prior to sending it to kilosort for spike sorting. And then the third step is calculating some QC metrics on the units that were pulled out. Each of those can have separate environment settings. Each of those can have different compute resources attached to it, but we can put them together into this pipeline. And again, this uses a tool called Nextflow, but again, my scientists don't have to learn how to write a Nextflow script, right? And so the real value that we see in this is that it allows us to build robust, reproducible pipelines, but lowering that scientific computing, data science, software engineering barrier for a lot of our scientists to be able to get these up and running and using them. So we've been developing a number of pipelines. Here are a few of them for spike sorting, for EFIS, calcium image processing. It uses Sweet2P, and light sheet imaging for some of the anatomy that we're doing. But one of the great things is we've built these on this platform, but all of the code for these pipelines, as pipelines, are in GitHub repositories that can be deployed outside of the CodeOcean platform, right? They can be deployed on your local machine or sent to your Slurm cluster at your university. So they're still, they can be used in a variety of contexts. All right, I'm going to jump back to talk about metadata and the kind of importance and value of metadata and how we're tackling that in our platform. Metadata goes hand in hand with file formats, and having data in standard formats allows us to build tool chains such as what Ben was talking about, that you can work really easily with the data. We find that while these standards are really good for data organization, they often do fall short on data documentation. And so we really need to have rich metadata to facilitate understanding and reusing the data by formalizing the embodied knowledge that enables people to actually make decisions about it. And if you go and look at like the guiding principles for like FAIR, the findable, accessible, interoperable, reusable, if you go look through that, every single one of them, there's like 12 bullet points. Every single one of them calls out the need for rich metadata to make data actually fair. So we've been developing a metadata schema that we use to document our data as we collect the data. And the schema documents a whole range of things. I'm just going to kind of briefly describe it. We start by describing the subject. All of our work is in mice, but our schema allows different species, but my examples are always going to be mice because that's what we work with. We want to know the species. We want to know things like the genotype, the sex, the age, the housing conditions, like the light cycle that the mouse lives in, any like things like that, as in terms of how and here, because we breed our own mice, we keep track of some of that breeding history of the subject as well. We keep track of the procedures that had been done to the subject prior to data collection. So this is an example of a mouse that had some stereotactic surgeries. A couple of surgeries were injecting viruses. We want to know what the virus was, what its actual plasmid is, the lot number of the virus prep that was injected, the coordinates of where we injected this virus. It also had a fiber implant inserted. So we want to know the coordinates of that implant. We want to know details about the optic probe itself. We also had this mouse on water restriction as it was trained on behavioral tasks. We also keep track of the instruments we use to collect data. So in this case, it might be things like lasers and cameras, maybe some behavioral apparatuses, and keep track of what those instruments are, and then how they're being used when we acquire the data. What's the power of the laser? Where are the cameras pointed? We want to know what the behavioral task is that the animal is doing. What's the software that controls the code that controls that logic? How did the animal perform on those tasks? And this describes the data as it's acquired. Once that data is acquired, we start processing it. We're going to then keep track of the processing, how the fiber optic signals, how we extract the signal from the raw data. And then we also keep track of some administrative details: who collected the data, who funded the data, what are the licenses for reuse, those types of administrative information. And finally, we add a lot of quality control into our metadata, where we have scientists evaluate pieces of the data, and those evaluations live in the metadata. And so this is kind of the only piece that's really a living piece of the metadata, and we've been developing tools that facilitate this annotation. But this is really, really crucial so that people are able to make informed decisions about which data they want to reuse. And we evaluate the different components of the data independently, because you can imagine in an experiment like this one, maybe something went wrong with the fiber photometry, and that data is really crappy and nobody should bother looking at it. But the behavior task, the animal did the behavior really well. The camera monitoring, that data is a high quality. And so if you're asking a question that has to do just with the behavior or just the poses of the animal, that data is still really valuable for those questions, but it might not be valuable for questions where you're actually thinking about the signal that we tried to image. So this is one toy example. I want to point out, as I mentioned at the beginning, we collect a lot of different types of data, and so it's important that our schema allow us to describe not just this fiber photometry example, but lots of other types of experiments. And so here's another example of another mouse, because again, we just use mouse, but whatever. It also got viruses, but different viruses were injected. And then we perfused the animal and extracted the brain, and we expanded it, and we gelated it and cleared it. We applied antibodies, and we want to know which antibodies we used. And then we used this smart SPIM microscope with this giant custom-made objective to image the brain that we then processed, and we created some of these fun visualizations that we throw around everywhere. And so the schema allows us to document a wide range of different physiology and anatomy experiments. And one of the things that we've been really leaning into here is leaning into persistent identifiers, ontologies, and databases so that we can establish controlled vocabularies to describe important components of these experiments. So this might be things like NCBI for having consistent naming of our species. For mouse, we use the Common Coordinate Framework for the Allen Brain Atlas to describe the different brain structures, and we can swap in other atlases for other species as needed. We use AdGene to document the viruses that we use. Wherever we can, we want to link information to existing ontologies and existing databases in order to make it as interoperable and to be able to link the information across different experiments and across different modalities. We find that ultimately what we're trying to do with this metadata is be able to communicate to different people and collaborators about what these data are, but ultimately that's also serving to link the different modalities of data that we're collecting in order to really pull that information to understand the system. So jumping back, I've kind of highlighted just the two things on the far end of our the philosophy of our platform. Ultimately, it's a bit of a modular platform, right? You could lean into pretty much any one of these things independently. You can lean into NWB files, or you can lean into putting data in public repositories, or use the metadata without using any of the other components, right? They're all independent pieces. And I think that would be, I think that's good. I encourage you to. But I really do think that the magic and the opportunity comes from pulling these pieces together to create a shared space to work together and to collaborate. I think, one of the key things that we're trying to do in our approach to open science is to move away from this kind of producer-consumer model where we're creating a lot of data and then we're disseminating it and we're creating systems to kind of put it out in the world, but really trying to encourage a system of community collaboration where it draws people into a common space so that we can leverage shared tools to really accelerate the way that we're able to work together. So I will wrap up. I just want to thank the members of our scientific computing team who've been developing this infrastructure and building a lot of tools to facilitate the use around this.
Thank you. If anyone has, I guess, a burning question for Saskia, you can ask that. If not, we're just going to move into the discussion. Oh, okay. Eve?
But being an old lady, I have lived through any number of generations of software tools that get updated and changed, and something you knew and understood no longer works. Either because you've changed the computer or you've gotten Acrobat has decided to upgrade itself, and so all the things you knew how to do are no longer there. They're somewhere else, but you don't know where they are. Mm-hmm. And so it seems like every year, things that used to be relatively easy, were maybe not as powerful, but they were easy, have gotten harder and more time-consuming. And so I guess the question I'd ask you, as time goes on and you're forced to update things and revise things and add new things, how do you ensure that you don't end up with a tremendous burden on your users to learn something new that they used to know how to do, but they no longer can do because you've changed it?
Yeah, no, that's a great question. So I think there's a couple of pieces to it, and one is that, sorry, I'm trying to think of which the three thoughts collided at once in my head. I think the key thing is that to the extent that we can, we're trying to lean into common tools, right? And so we're putting, as I mentioned, like all of our neurophysiology data is going into NWB, and the way we access that is using PyNWB. And so the thing that we're trying to not do is create a separate layer of software that works specifically for the Allen Institute for Neural Dynamics data, but doesn't work for similar data that are being generated by other people, right? And this is why I think it's really valuable and really crucial that we lean into these types of community standards, and so that we can have these common tool chains. There is this piece, though, that as we integrate new experiments, because science is always moving forward, and as we integrate new experiments, there's going to be ways in which our existing tools fall short, right? Like our metadata schema, if we suddenly start doing like fMRI, that's not currently captured in my schema. Not quite, right? And so I'm going to have to add a new concept, but that concept isn't going to interfere with the current metadata, right? It's going to apply to new things. And so we have a way of, we have a system for version control on the metadata, and we are very careful when we update it to make sure that as infrequently as possible that we have to really affect other people. And there might come moments where we really do, because there have been enough changes, like we really do have to like revisit some of these things, but our goal is to make those very intentional and very infrequent when possible. But we can't stop science from progressing. And so what we're trying to do is really go in with open eyes and with a lot of intention to capture what we can, to lean into these community tools so that we're able to leverage that community effort. But then, you know, we do have to be realistic. But we're trying as much as we can not to. I hate learning new things too. So I'm kind of one of those people that stops people from changing things if we really don't have to.